Good afternoon, all of you. So now come to human genome project, right? So these subtopics, two subtopics are the human genome project and DNA fingerprinting. So the two topics comes under molecular basis of inheritance in previous chapter, right? So in previous chapter, we not discussed right DNA fingerprinting and human genome project. So that two topics. So now we are going to discuss, right? So come to first subtopic, that is human genome project. What it is? Human genome project. So what is human genome project? So what is the goal? What are the major goals of human genome project? And what are the advantages of human genome project? So what is human genome first? Right? So we learn all these things. So genome first. What is genome? Definition for genome: complete set of genes or genetic material which is present in cell or organism called genome, right? So what is genome here? Definition for genome: complete set of genes, complete set of genes or genetic material, complete set of genes or genetic material in in a cell, in a cell or organism, in a cell or organism called genome. So is it clear? Definition for genome: so what is genome? Complete set of genes or complete genetic material which is present in a cell or organism called genome, like that. What about human genetic material or human genome here? So human genome also same thing, complete set of genes or genetic material which is present in human cell called human genome, right? Then, so the development of molecular techniques, development. So the development of molecular techniques, molecular techniques for analyzing genes, for analyzing for analyzing genes and gene expression, genes and gene expression starts a revolution in experimental biology. See, come to second point here. So the development of molecular techniques. So that molecular techniques developed right in the year 1952. So now, right, that molecular techniques developed, right? So that molecular techniques, development of molecular techniques for analyzing genes, analysis of genes and gene expression starts a revolution in experimental biology, right? So once DNA sequencing was started, once DNA sequencing, right? So once DNA sequencing started, so once DNA sequencing was started, see, geneticists and other biologists and other scientists realized that to determine entire sequence of genomes of other organisms, right? So listen carefully. Once DNA sequencing techniques or DNA sequencing was started, see, genetic scientists and other biologists realized that to determine entire genomes of other organisms. So what is genome, sir? So what is sequencing, sir? So genome is nothing but a complete set of genes which are present in that genetic material, or complete genetic material which is present inside the cell called genome, right? So when DNA sequencing techniques were started, so that genetic scientists and other biologists starts otherwise realized that to determine the entire genome sequencing of other organisms, it means so that other organism genome sequencing, right, also possible realized by the other scientists once DNA sequencing techniques were started, right? So what is sequencing, sir? Don't confuse. The DNA sequencing is nothing but, so how that nitrogen bases are nucleotides, and what type of nucleotides located in that genetic material, how they are located in that genetic material. So first, what type of nitrogen base will come? After that, what type of nitrogen base will come? A, Z, C, T, right? So like that, how that nitrogen bases are nucleotides located in that genetic material. What is the actual sequence of that nitrogen bases in that genetic material? So that is called DNA sequencing. DNA sequencing. DNA sequencing is nothing but how that nucleotides or how that nitrogen bases located in that genetic material to determine nitrogen bases location. Otherwise, how that nitrogen bases are nucleotides located in that genetic material called DNA sequencing, right? So once DNA sequencing techniques were started, so genetic scientists and other biologists also realized that to determine the entire genome of other organisms also possible. Other organisms genome sequencing also possible, realized by the other scientists also, right? So sequenced first sequenced genetic material is, so first sequenced, right, complementary sequenced genome. So first complementary sequenced genome is mitochondrian genome. Human mitochondrian genome. Mitochondrian, singular mitochondrian, right? Plural mitochondria. So mitochondrian genome, right? So which genetic material first sequenced? So that first sequenced genome or genetic material is mitochondrial genetic material or mitochondrial genome. So this mitochondrial genome includes around 16,159 base pairs, right? So how many base pairs present in that mitochondrial genome or mitochondrial genetic material? 16,159 base pairs, right? So this technique, otherwise this genome sequencing technique, right, so completed in the year 1981. Right? So when they discovered that human mitochondrial genome sequencing in the year 1981. So that human mitochondrial genome consists of how many base pairs? 16,159 base pairs present in human mitochondrial genome or genetic material, right? Then see, what about nuclear genetic materials or human nuclear genetic material? So human nuclear genome larger than mitochondrial genome, right? 2 lakhs times larger than mitochondrial genome. See, no doubt, human nuclear, nucleus or nuclear, your wish, nucleus genome larger, how much larger, right? So around 2 lakhs times larger, 2 lakhs times larger than mitochondrial genome, larger than mitochondrial genome, right? So it means compared to mitochondrial genome, right, that nuclear genome is larger, right? Approximately 2,000 times larger than the mitochondrial genome, right? Then, so now come to here, what is advanced techniques? See, this human genome project also associated with a recently developed science or a rapidly developed area, that is bioinformatics. So what is bioinformatics now? See, that human genome project also associated with a recently developed area or rapidly developed area, that is bioinformatics. So what is bioinformatics? So see, study of biological information with the help of computers called bioinformatics, right? So computerized programs, with the help of computerized programs, So we will study biology, right? That type of branch is called bioinformatics, right? So definition for bioinformatics: study of biology, study of biology with the help of computers, computerized programs, otherwise, with the help of with the help of computerized programs, computerized programs, or computers, otherwise, simply. See, what is bioinformatics here? See that human genome project, right? Closely associated with recently developed area, right? That is bioinformatics. What is bioinformatics? Study of biology. So with the help of computerized programs called bioinformatics, right? So and one more branch, that is genomics. Genomics. So what is genomics? So development applications of mapping, sequencing, and computerized procedures to sequencing of entire genome of organism called genomics, right? So write down definition for genomics. So development applications development applications of sequencing, mapping, sequencing, and computerized procedures. Computerized procedures, right? Computerized procedures for analyzing, sequencing of, sequencing of entire genome of organism called genomics. Important definition for genomics: development applications of development applications of mapping and sequencing and computerized procedures, right, for sequencing of entire genome of organism, right, for analyzing or for sequencing of entire, right, genome of organism called genomics. Is it clear? Definition for genomics: development applications of mapping, sequencing, and computerized procedures, right, for analyzing entire genome of organism called genomics. So what is mapping? So here it is mapping, again two types, right? Physical mapping, genetical mapping and physical mapping. What is genetical mapping? It means where exactly genes are located in chromosome, right? What type of genes located in particular chromosome, right? So how much distance is present in between that two genes? So those genes are useful or not useful genes, right? So how they are located, exactly where they are located in that particular chromosome. So we call it as mapping, actually, mapping. And what about sequencing? So sequencing is nothing but how the nucleotides located in that genetic material, right? What type of nitrogen base is present in that DNA, right? So that information comes under sequencing or sequence. Then computerized procedures, right? It means we have to develop some softwares, right? Some programs, computerized programs for analyzing that sequence. It is not possible to count that nucleotides which are present in genetic material, right? So with the help of computerized programs only, it is possible to determine that sequencing of genetic material, right? So now come to definition for genomics: development applications of mapping, sequencing, and computerized procedures for analyzing entire sequence of entire genome of living organisms called genomics, right? So there are three subfields again in genomics. One is structural genomics, structural genomics, structural genomics. Second one, functional genomics and comparative genomics. So first come to structural genomics. Sir, what is structural genomics? So see, the structural genomics includes genetical mapping, genetical mapping. Then second one, physical mapping, physical mapping, and third one, and sequencing, right? Physical mapping, genetical mapping, and sequencing. That structural genomics deals with, otherwise includes, genetical mapping, physical mapping, sequencing of genome in organism, right? So structural genomics include genetical mapping, physical mapping, and sequencing of entire genome in an organism called structural genomics, right? Then what is genetical mapping? So see how genes are located in chromosome, where exactly they are located, called genetical mapping. Physical mapping, so what is the distance in between the two genes, right? So what is the actual distance in between the two genes, right? Explains physical mapping. Then sequencing is nothing but how they are located, right? Then come to second field, subfield in genomics, functional genomics. Functional genomics. Functional genomics. So what is functional genomics? So name like the information, functional genomics. Yes. So this functional genomics includes what are functional genes, what are non-functional genes, right? It explains genes and non-genes. Genes and non-genes means what are functional genes, what are non-functional genes. Functional genes and non-functional genes. Non-functional genes, right? So it explains or it deals that functional genomics deals that. So what are functional genes, what are non-functional genes? So that information explained by the functional genomics. Then another area, third area, that is what it is, third area, comparative genomics. Comparative genomics. So I'm writing here comparative genomics. Third one, comparative genomics. See that comparative genomics deals that genome information comparatively in between organisms, right? So this comparative genomics deals comparison of genomes, comparison of genomes of organisms, genomes of organisms, right? So these three subfields comes under genomics branch, right? So don't confuse what is genomics means. Development applications of mapping, sequencing, and computerized procedures, right, for analyzing gene sequencing in cell or organism called genomics. It includes three sub branches. So structural genomics, functional genomics, and comparative genomics, right? Structural genomics explains genetical mapping, physical mapping, and sequencing. Then functional genomics explains what are functional genes, what are non-functional genes. Comparative genomics, so comparison of genomes in between organisms, right? That it's a brief introduction, right? Human Genome Project introduction. Take on screenshot now. Right. So now come to right. So some other information related to Human Genome Project. So it was mega project actually. So that Human Genome Project is a mega project. Mega project. So this one of the main partner in that mega project is Wellcome Trust in USA. Wellcome Trust is the major partner. Wellcome Trust actually, one trust name, right? Wellcome Trust is the major partner in Human Genome Project. Human Genome Project, right? Human Genome Project. Okay then. So it was mega project. Actually, they planned to start Human Genome Project in the year 1986. So many states proposed, right, that Human Genome Project proposed in the year 1986. So that Genome Project proposed in the year 1986, but it was started. It was started in the year 1990 October. So 1990 October, that Human Genome Project started in the year 1990 October. See, they plan to complete this Human Genome Project. They planned to complete this Human Genome Project within 15 years. So they planned to complete that Human Genome Project within 15 years. 15 years. But due to advanced technology, that Human Genome Project completed in the year, see, 2003. 2003. So it completed due to advancement or advanced technology. It completed April 14th, 2003. Within 13 years, so it is completed, right? That Human Genome Project. So it is also associated with that Human Genome Project is also associated with. It is also associated that Human Genome Project is also associated with Department of Energy. Department of Energy, DOE, Department of Energy, National Institute of Health, National Institute of Health, Human Genome Program, HGP, Human Genome Program, HGP, right? And Human Genome Research Institute, right? So National Human Genome Research Institute, right? See, this Human Genome Project is also associated with the Department of Energy, National Institute of Health, and Human Genome Program, and National Human Genome Research Institute, right? So these organizations contribution also there in Human Genome Project, right? It is also associated with DOE. What is the DOE? Department of Energy, and NIH, National Institute of Health, Human Genome Program, right? National Human Genome Research Institute, right? Then see, along with US, other states contribution also there. Other states contribution also there in Human Genome Project. Along with US, Britain, Great Britain, Japan, China, and other countries contribution also there in Human Genome Project, right? So along with US, United States, Great Britain, Japan, China, and other countries contribution also there in Human Genome Project. So now come to goals of Human Genome Project. Goals of Human Genome Project. So what is the main goal, or what are the main goals of Human Genome Project? So first important goal is identification of protein encoding genetic material or protein encoding genes which are present in human genetic material, right? So right now, first goal is identification of protein encoding genes. Protein encoding genes which are present in human DNA, approximately 20,000 to 25,000 genes. 25,000 genes which are located in human DNA. Human DNA, right? So first important goal of Human Genome Project is identification of protein encoding genes which are present in human genetic material DNA. But in our material they given 30,000 genes. 30,000 genes, but NCERT is final. In NCERT, they given approximately in between 20,000 to 25,000. Better to take 20,000 to 25,000 only, right? Then, first important goal of Human Genome Project is identification of protein encoding genes which are present in human genes or human genetic material, right? Then, second one, determination, determine, determine, or determining. So, determining 3 billion bases, 3 billion bases, 3 billion bases or 3 billion nucleotides which are present, are that makes DNA, that makes human DNA, right? See, to determine 3 billion bases which are involved in the formation of human genetic material DNA. This is second goal of Human Genome Project. See, approximately 3 billion bases are present in that genetic material, no, right? To determine that 3 billion bases that which participates in human genetic material is second goal of Human Genome Project. Then third goal again. So obtaining information preserved in the form of data. Obtained information, all right? Storing obtained information or collected information in the form of data, right? So that collected information or obtained information preserving in the form of data. It is another important goal. Third important goal, storing obtained or collected information in the form of data. Then one more again. So developing tools for data analysis. Developing tools for data analysis. Tools for data analysis. So developing tools for data analysis. Then transferring this technology to the private sectors. Another goal, transferring this technology to the private sector. Like industries. Next goal is transferring this technology to the private sectors like industries. And one more goal, addressing ethical, legal, social issues which arises in Human Genome Project. Addressing ethical, E means ethical, legal, social issues, legal and social issues which arises in Human Genome Project. Social issues which arises in Human Genome Project. These are important goals. So what are the major goals in Human Genome Project? So identification of protein encoding genes which are present in human DNA, approximately identification of 20,000 to 25,000 genes, right? Protein encoding genes which are present in human DNA, right? So second one, determining 3 billion bases which participates in the formation of DNA molecule, right? Then third one, storing that obtained information or collected information in the form of data in computers, right? Then so developing tools for data analysis and transferring this technology to the private sectors like industries, then addressing some ethical issues which arises in human genome project, and legal issues which arises in human genome project, then social issues, right? So addressing ethical, legal, social issues which arises on human genome project. So these are the major goals of human genome project, right? So it is a mega project. One of the major partner in that is Wellcome Trust in USA. Then it was proposed in the year 1986, but it was started in the year 1990 October, and they plan to complete this human genome project within 15 years. But due to advancement in molecular biology, so they completed April 14th, 2003. Within 30 years, they completed, and it is also associated with some other organizations or branches, right? Department of Energy, National Institute of Health, and Human Genome Program, and National Human Genome Research Institute, right? So these organization contribution also there in human genome project. Then along with US, so Great Britain and Japan, China, and other countries contribution also there in human genome project, right? Then these are the goals. Take a screenshot. What are the major goals of human genome project? Important. Important goals of human genome project. Then now, right, comparative study of human genome. Now come to comparative study of human genome. Comparative study of human genome. See, along with human genome, some other organisms genome also sequenced. See, comparatively, along with human genetic material, other organisms genetic material also sequenced, right? So they identified other organisms genome sequences also. For example, you can take E. coli, Escherichia coli. Escherichia coli. So that Escherichia coli genome consists of 4.64 MB megabases 4.64 megabases. And second one, Saccharomyces cerevisiae. So what is Saccharomyces cerevisiae? Yeast. Right. First one is bacteria. So that E. coli bacterial genetic material or genome consists of 4.64 megabases. But what about Saccharomyces cerevisiae? Yeast actually. Yeast, fungal member. So that to unicellular fungal member Saccharomyces cerevisiae. Baker's yeast or brewer's yeast. Right. That yeast also unicellular. So that genome consists of 12 lakhs 67,280 base pairs. 280 base pairs, not megabases here. 280 base pairs. So 12 lakhs 67,000, 280 base pairs, right, present in genome of Saccharomyces cerevisiae. What is Saccharomyces cerevisiae? Yeast actually. Yeast, unicellular fungal member, right? Then Canarobditis elegans, another one, nematode. So 16 chromosomes present in Saccharomyces cerevisiae. That 16 chromosomes genome consists of 12 lakhs 67,280 base pairs. In the same way, another organism, Canorhabditis elegans. So Canorhabditis elegans. So that Canorhabditis elegans is a nematode actually. Nematohelminthes organism are nematode. So that genome also sequenced, it consists of 100.3 megabases, MB, megabases. 100.3 megabases. Then Drosophila melanogaster, another organism, Drosophila melanogaster. Okay, Drosophila melanogaster is also known as a fruit fly, simply fruit fly. So that fruit fly genetic material also sequenced, right? So it consists of 118.4 MB megabases. 118.4 megabases. So what it is? Drosophila melanogaster, fruit fly, insect. Then next one, mus musculus. So mus musculus, right? What it is? Mouse. So in mouse, 99% of the genes, 99% of the genes are related to human genes. So 99% of the functional genes are related to human genes in mus musculus, right? Then functionally related to humans. Mus musculus is nothing but mouse. Then Arabidopsis thaliana, Arabidopsis thaliana. So that Arabidopsis thaliana is one of the plant actually, angiospermic plant, flowering plant. That Arabidopsis thaliana also consists of, that genetic material or genome consists of 120 megabases. 120 megabases, right? So comparative study of human genome with other organisms, right? So when that scientist sequencing that human genetic material, at the same time they also sequenced other organism genetic materials, right? So like E. coli, that E. coli genome consists of 4.64 megabases. Then Saccharomyces cerevisiae yeast genome 12,67,280 base pairs. And Canorhabditis elegans, one of the nematode. So that Canorhabditis elegans nematode also genome also consists of 100.3 megabases. Then Drosophila melanogaster consists of 118.4 megabases. Then mus musculus, 99% of the genes, functional genes similar to the humans. Then Arabidopsis thaliana, 120 megabases, right? So comparative study of human genomes, right, with other organisms. So now come to methodologies again. Methodologies. What about methodologies? So there are two methods or two approaches for sequencing of genetic material or genome, human genome, right? So they are, one is expressed sequence tags. So second one is sequence annotation, right? So there are two major approaches. There are two major approaches for sequencing of human genetic material. In that, first one is expressed sequence tags. Expressed sequence tags. Expressed sequence tags. Sir, what are expressed sequenced tags? So within the brackets, you have to write ESTs, expressed sequenced tags, right? See, the genetic material which is expressed as RNA molecule called expressed sequence tags, right? So in that genetic material, in that DNA or genetic material DNA, so the segments which participates in transcription process, so those DNA segment sequences only they identified, right, by using this technique, expressed sequence tags. What are expressed sequence tags means? See, the DNA which is expressed as protein, expressed as RNA, otherwise simply RNA, either it is mRNA or rRNA or tRNA, whatever it may be. So the DNA molecule which is expressed as RNA molecule, right, called expressed sequenced tags. So that expressed sequences information only they identified in this process, right? So we call it as transcript units. So the transcript units. So the transcript units information or that sequence information only identified in this approach, right? So we call it as the expressed sequenced tags. So some part of genetic material information only sequenced in expressed sequence tags, right? Then second method again, second approach. So sequence annotation. Sequence annotation. Sir, what is sequence annotation? So see, in sequence annotation method, first they isolated genetic material, so by using various enzymes. What type of enzymes already we discussed, right? So in sequence annotation method, so first isolated genetic material DNA in first method. Second method, so fragmented genetic material by using restriction endonuclease enzymes. Fragmented genetic material by using restriction endonuclease enzymes. Second method, right? Then again, they separated the DNA fragment, desirable DNA fragments, right? They separated and inserted into vector. Into vectors. Right, in this method, so that genetic material DNA fragments separated and inserted into the vectors. So what type of vectors they are using in this method? So BAC and YAC. What is BAC and what is YAC? BAC means bacterial artificial chromosome. It is nothing but PBR 322. Is YAC, East artificial chromosomes, right? See, in sequence annotation method, first they isolated genetic material DNA, again they fragmented the genetic material. So those fragments inserted into the vector. Which type of vectors they are using? Bacterial artificial chromosome and East artificial chromosome. After that, they amplified the genetic material. So after amplifying that genetic material by using BAC and F, bacterial artificial chromosome and East artificial chromosome, so that segments information completely sequenced by using computerized programs, right? Automated DNA sequencing techniques, right? See, after cloning, so that segments or fragments sequenced by the automated DNA sequencing techniques. So that fragments sequenced by the automated DNA sequencing technique, by automatic DNA sequencing, automated DNA sequencing technique. So that automated DNA sequencing technique is computerized programming, right? So with the help of computerized programming, that is automated DNA sequencing technique, so easily we'll identify that fragment sequence. Like that, each and every fragment which is present in that genetic material sequenced by the automated DNA sequencing technique. So it is computerized program, actually, automated DNA sequencing technique, right? That automated DNA sequencing technique first explained by the Frederick Sanger. right? Frederick Sanger, right? Who developed automated DNA sequencing technique? Frederick Sanger. Frederick Sanger developed automated DNA sequencing technique. So that by using that automated DNA sequencing technique, so that DNA fragments sequenced, complete DNA fragments sequenced, and again, so that information printed, otherwise that information preserved in the form of data in computers. So that chromosome, each chromosome consists of many segments or many fragments, right? That fragments information sequenced and preserved in computers. So for example, first chromosome, so consists of this type of sequence, and again second chromosome, third chromosome, fifth, like that 46 chromosomes information also preserved in computers in the form of data by using automated DNA sequencing techniques, which was developed by the Frederick Sanger. Right, Frederick Sanger. See, there are two major approaches for sequencing genetic material DNA. One is expressed sequence tags. Second one is sequence annotation. In expressed sequence tags, so the DNA which is expressed as RNA molecule only sequenced, right? So we call them as transcript units. Transcript units only sequenced in this process, right? In second method, sequence annotation. So they isolated genetic material, fragmented that genetic material, that fragments inserted into the vectors, bacterial artificial chromosome and yeast artificial chromosomes. They amplified that genetic material. So that amplified genetic material sequenced by using automated DNA sequencing technique, which was developed by the Frederick Sanger. So after developing that sequence, so they preserved that information in the form of data in computers, right? Right. So note down this methodologies: comparative study of human genome with other organisms and methodologies, expressed sequence tags and sequence annotation. Don't confuse. There are two methods for sequencing of genetic information, but in this process or in this method, just the DNA which is expressed as proteins, which is expressed as RNA molecules, so that information only we sequenced here. But here complete genetic material, 2.2 meters genetic material, right, sequenced in second method, sequence annotation method, right? Then small siding, salient features of human genome project. Salient features, important features of human genome project. So that important features are salient features of human genome project, and what are the advantages of human genome project. So that information we'll see in tomorrow class, right? So just I give an introduction about human genome project, when it was started, when it was completed, right? That information only I given in this. So now salient features of human genome project and applications are advantages of human genome project information we'll see in next class.